Já. Estão todos desligados, acho que é lá em cima. Estamos aqui com... Good morning again. Let's continue with the second section of this panel on climate change and infectious disease. After coffee, we have 4 presentations. Luiz Alcântara, we start with the expanded navigation project for intensive and optimized surveillance, the NAVIO project. Then Maria Ogrewalska, she will present her work on environmental and animal surveillance in Antarctic areas. Follows Juliana Calabria. The title of her presentation is Surveillance of Pathogens in Sewage from Hospitals in Belo Horizonte and Campo Grande and in wastewater at the Paraguay River in the Pantanal Biome. And RaoniRajão, his talk will be on impact of deforestation and greenhouse gas emissions on climate change. So I'm very pleased to introduce the first talk. Luiz Alcântara is one of the coordinators of the International Climate Consortium and coordinator of the Virus Evolution and Molecular Epidemiology Bioinformatics workshop. He is a senior researcher at the Oswaldo Cruz Foundation, RaoniRajão, Fiocruz-Minas Gerais, and a collaborating researcher at CTVacinas. He holds a PhD in cell and molecular biology from Oswaldo Cruz Foundation. He's also completed 2 postdoctoral fellowships, one at the Nelson Mandela Medical School and the University of KwaZulu-Natal in South Africa, and another at the National Cancer Institute. He is a member of the Arboviral Surveillance Networks, RELDA, and Vigenda, and a pathogen genomic network of the Pan American Health Organization. Luiz, we are very pleased to have you here. The floor is yours.
Okay, thank you to be here, and it's a pleasure to present our project NAVIO. And as I have many slides, I have to show some slides, other not. And then I will speak, I think the majority of them in Portuguese, okay, to be more realistic to related to this project. And as Sonia told you, this is the NAVIO project, expanded navigation for optimized and intensive surveillance. And This project is in collaboration with the Brazilian Navy and also with the Health Secretary of the states of Mato Grosso and Mato Grosso do Sul, and many others partners. I don't have space to put all the partners here, but I know that there are other partners here present. And this is part of the Climate Consortium also. And up to now, I had the fund from NIH, for National Institutes of Health, and from the PAHO, the Pan American Organization, and also from CNPq, from NIH stopped. Trump stopped the project now, but I'm trying to other funding and to be able to continue with the project. This is a project to see how the climate change could intensify the diseases, and also to increase the diseases, it to increase also the pathogens. It's before was only the arboviruses and the respiratory virus, but you have to change because the reality in the community is for all kinds of diseases. You have to integrate other people that work, other research that work with leishmaniasis, Chagas diseases, many other diseases, and you can see here. No, it's up. I don't know. This is some movies, but it's not working. And our travel, we started here in Corumbá Ladário. Here is when you set up the Navy stay here, and then you go to 2 trips. One trip to the south of the river. This is the Tramo Sul trip. And you have some communities in this area. This is a trip for around 21 days. And you have another trip going to the north of the river, up to Cáceres and Mato Grosso. And you have here many others riverside communities. And the project is to see how the climate change the diseases, increase the diseases. And you have all these partners here. You have the support for locals, for example, that put in the ship many equipment that was be able to have one ship with a laboratory for serology, for molecular biology, another laboratory for environmental surveillance, another one for vector surveillance, and also for animal sample surveillance. And you have also now with Biomanguinhos. Biomanguinhos give us the support to have a laboratory for biochemistry, all biochemistry tests, and also for hemogram. Tá, o próximo passa. And here you have one video. It's just 5 minutes of the video that give you all the reality. How is this NAVIO project? Sorry, because it's in Portuguese.
E as alterações no clima têm a ver com as doenças. Segundo os pesquisadores, inundações, secas extremas e aumento médio da temperatura impactam diretamente na distribuição dos transmissores e dos hospedeiros de doenças infecciosas. A maior incidência do mosquito Aedes aegypti numa região, por exemplo, facilita a propagação de dengue, zika e chikungunya. A Fundação Oswaldo Cruz e a Marinha, com o apoio do Ministério da Saúde e de outras outras instituições estão descendo o rio Paraguai para levar atendimento aliado à pesquisa às comunidades ribeirinhas. Na triagem, as técnicas e enfermeiras fazem o acolhimento e já iniciam o levantamento de dados.
A gente procura saber como é que eles estão vivendo, quais são as características dessa população, quais são os principais problemas de saúde, avalia a caderneta de vacinação e ao identificar algum problema de saúde a gente faz os encaminhamentos.
Enquanto os ribeirinhos recebem atendimento, outras equipes percorrem a região para conferir as condições de vida deles. Foram recolhidas amostras da água do rio e das casas dos moradores.
Nós conversamos com eles, como eles fazem com as captações de água, como eles armazenam a água, se realiza algum tipo de tratamento. Então, nós coletamos a água que eles ingerem e também que são utilizados ali para os afazeres domésticos.
Também foi coletado material genético dos animais domésticos.
A tecnologia do laboratório empregada no projeto tem condições de identificar RNA viral também das fezes coletadas. Então, a gente aproveita todas as oportunidades e coleta também esse material.
Os navios foram adaptados para este projeto. Aqui, por exemplo, onde funcionava um alojamento, foi montado um laboratório de alta tecnologia com a ajuda de parceiros. São mais de 3 milhões de reais em equipamentos que fazem desde um hemograma até um sequenciamento genético, que permite identificar o tipo de vírus e as variantes que estão circulando na região.
Nos permite entender melhor os micro-organismos que causam doenças e que estão circulando na região, além de permitir também a identificação de novos micro-organismos. Essas duas abordagens de sequenciamento podem nos ajudar a propor ações de saúde pública.
Louela trouxe da Holanda tecnologia para identificar 24 arbovírus diferentes em 64 amostras de sangue ao mesmo tempo. Ela diz que o método é mais eficaz que os testes individuais e traz mais agilidade.
Quando você faz o teste individual, pode haver uma reação cruzada. Pode haver um falso positivo, às vezes para dentro, por exemplo, e na verdade é Zika. E com esse teste, analisa os anticorpos para todos os vírus ao mesmo tempo. E tem um padrão para cada reação para cada arbovírus. Em cada ponto branco que está na lâmina, está impresso para analisar 24 arbovírus. Esta lâmina tem ao todo 64 quadrinhos que dá para analisar 64 pacientes ao mesmo tempo.
Já a Margarida representa uma universidade da Alemanha, parceira do projeto. Ela pesquisa bactérias resistentes a antibióticos.
Bacterias que são multiresistentes son una gran problemática actualmente en hospitales porque los pacientes ya no pueden ser tratados.
Esta é uma das atividades de coleta mais difíceis de fazer. Com o apoio da Marinha para dar segurança, os pesquisadores vão se embrenhar no mato para capturar mosquitos. E é claro, não pode usar repelente, né, professor?
Verdade, indispensável usar roupa bem protegida, bem grossa para os mosquitos não picar e perneira por causa da picada de cobra, claro.
Agora, como vocês fazem essa coleta?
Nós fazemos coleta quer com um aspirador que suga os mosquitos por este cano e são acumulados aqui, quer com um pulsar para coletar mosquitos mais em voo e depois são sugados com este aspirador bucal e depois são levados para o navio.
É uma tarefa demorada. Os mosquitos vão para o laboratório embarcado. Começa a segunda etapa desta pesquisa.
O grande diferencial desse projeto é a resposta rápida, né? Nós coletamos o mosquito agora em campo. Dentro de meia hora, mais ou menos, a gente já consegue saber a espécie de mosquito. Então, por exemplo, se eu tiver a Aedes aegypti, que transmite dengue, e coloca essas populações que vivem em áreas isoladas em risco, dentro de meia hora a gente consegue dizer que tem esse mosquito, que essas populações poderiam estar em risco e com um pouco mais de tempo, no máximo até o fim do dia, a gente consegue dizer até se tem vírus nesse mosquito.
O projeto Navio tem duração de 5 anos, podendo ser renovado por mais 5. Os 2 primeiros anos de pesquisa trouxeram resultados significativos sobre os vírus que circulam no Pantanal.
Nós encontrávamos 2 mosquitos, 3 mosquitos de cada espécie. Hoje a gente está encontrando bastante. Estamos encontrando também muitas pessoas com sintomas relacionados à chikungunya, tá? Que são vírus urbanos que já estão aqui, e o que faz esses mosquitos urbanos mudarem para regiões como essa silvestre, já que é urbano, porque está aumentando a temperatura que está se igualando à temperatura existente em áreas urbanas.
Esse mapeamento ele serve para poder conhecermos, né, o que circula, quais as doenças, os vírus e bactérias que circulam dentro do nosso estado, junto com essa população, e para que a gente possa programar, através de políticas públicas, né, e trazer um atendimento melhor para essa população.
É a ciência a serviço da população mais vulnerável.
Todo esse movimento que eles fazem para estar aqui com a gente, tão longe que eles vêm, largam suas famílias, e lembra que a gente está aqui no fundinho do mundo aqui, mas a gente é importante para eles e eles também são importantes para nós. Por isso a gente agradece eles estarem aqui.
E as alterações?
Ok. This is the workflow of our trip. Né? You can see here 3 ships. In one ship you have there are the consultory for physician and also for dentists, and the others 2 you have the laboratories that was set up there. And as normally you stay 3 days in each community. Né? When you arrive in the first day, you go to visit the house of the families, the riversides, and there you give you explain about the project. You start doing make the health education in these places, and also invite them for another day to go to in front of the ships. Né? In front of the ships, you have some tents, and these tents, one of the tents is they are waiting here, and you also start here the health education, and here you have the trial of the riverside families, né? And you have in the red cap all the data. You collect all the data, including the red cap here in the tablet, and all this information goes to the doctor inside the ship, because they also have their own computer or a tablet with the information. And as soon you collect all the information in the questionnaire, this information, the riverside are sent to other tent to collect the sample. You collect the blood samples, you collect the feces samples also from them, and also you collect the oropharyngeal sample also here. This sample are sent to the labs that is inside the ship. You have here the mini LACEN. LACEN is the central laboratory of the state of Mato Grosso and Mato Grosso do Sul, work together with all the equipment necessary to make the serology and molecular biology assays. And other samples are sent to the feces are sent to the lab to make the tests also. And after this, they go to the physician. Normally you have 2 physicians from the NAVIO, and sometimes you also include other physicians from different universities, like Federal University of Minas Gerais, is a physician more specialist and other diseases. And after this, they go to the dentist. You have 2 dentists inside with all the equipment necessary. And for the last step in this day, the second day, is the vaccination. You have all the vaccine for the immunization program from Brazilian Ministry of Health inside the ship. And you have all the vaccines. And in the 3rd day, they come again to the doctor. All the exams was finished. As soon as the exams are finished, the result are sent to the physician. The physician attend again the riverside, and give the result and also the prescription of the medicines for them. You have also a pharmacy inside and with all the medicine that is necessary, because the ship was included in as the reference ship and helping the riversides in the SUS. SUS is from Brazil. The Brazil knows about the SUS. And then, because all the municipalities send the medicines to the ship. Then this is the third day. You end again with one section for health education again, based in the result that was found during the trip. And this is our workflow. This is all the vaccines that you have inside. You are having the coverage is high now in the riverside. The majority of them want to be vaccinated. Of course, to do this, you need to do a good education related to vaccine with them. And this is the route that I told you. What you are found there? Juliana Calabria is responsible for the environmental surveillance. She will present the result about this, because she will present today. But one thing that I want to say, that in the water of the river, you have 100% of E. coli. E. coli in these samples that was collected. You also analyzing, you have another research from the project, Jonathan Abraham, that will be here tomorrow, that will show the giant virus that are being collected there, because giant virus is a good market for change of to increase the temperature in the region, because increasing the temperature increase also the presence of acanthamebas, amoebas not pathogenic, and if you have more acanthameba, of course you have more giant virus that stay inside these acanthamebas. He is doing this. You can see also in the drink water, the quality of drink water, with 76% of total coliforms and also E. coli in the houses. And also in the parasitological analysis, you can see 72.2% of the with the pathogenic parasites in these people, more than 50%. And other thing that you are seeing here is because you went 2 times during 2 years, 1 time in each term south and term north. And in the term south, going to the south of the river, you can see here the number of mosquitoes that was collected. This is the number. If you see in the year 2, is the number of mosquitoes. You can see between 2 years, you can see increase a lot the number of mosquitoes in this area. Also here in the north, in the first year was this, almost 700 mosquitoes, and now more than double of mosquitoes. And you also doing the animal surveillance. All the samples for mosquitoes are going to metagenomics, are being prepared for metagenomics, and also the samples, the animal samples, metagenomics and also metatranscriptomics. And you can see here the rapid test. It's impossible in this time to say all the rapid tests that you have here, but I can show you that just this, that is very important. The number of cases of syphilis, many cases of syphilis. You have around 8% of syphilis in these areas. In the year 1 and also in the year 2. Oh, sorry. Year 1 and year 2. You can see the case of syphilis. The case of syphilis. Here is the result for the molecular tests, the PCR, during the year 1 in the south, year 2 in the south also. You can see this is panel for respiratory virus. The majority of the respiratory virus that you are found is the rhinovirus, and it's also here during the year 2. And in the north part of the river, you have also this is the panel, also rhinoviruses, here and here during the trip 2. And you also perform the molecular diagnostic for arboviruses. In the south part of the river, you did not detect it. No, and you didn't have acute case for arbovirus. Of course, you didn't detect nothing of arbovirus. And also here in the north, it's a little bit, you have just chikungunya in the year 1 and dengue 2 and the year 2. Then metagenomics, you are doing metagenomics and genomics inside the ship. You have a laboratory for genomics and metagenomics. You use the platform for nanopore, for Oxford Nanopore. And then you are doing this. You are sequencing respiratory virus, bacteria that are related to respiratory symptoms, metagenomics for mosquitoes also, and sequence all the cases for example, here is dengue. It's one person, one riverside that was HIV positive, but was also with symptoms for arbovirus, and then was dengue 2. Here for rhinovirus, this one example for rhinovirus. You did also the analysis, all the bioinformatic analysis, the evolutionary analysis. Aqui also enteroviruses. You are found HTLV, HTLV1 and HTLV2 in the population. This is in the south. We started this in the second year. This is in the south. Going to the south in year 2, this is one man that have, this is they have this problem here. You take in the project one dermatologist. You also performed the HTLV. He was positive for HTLV1. And in the north of the river, you went in the year 1. You have two cases, one in Barra de São Lourenço and Mato Grosso do Sul, another one in Porto Carne Seca in Mato Grosso. And you have in our last trip that was in the north area, in the year 2, you have 5 cases for HTLV. This is very important because as soon as you have the result, you send to the health secretary of the states the reporter, and then they will make active surveillance in this area. And also, all the results are notified in the database of the Brazilian Ministry of Health, that is the GAL and the SINAM. You do this, everything. This is the result for the serology. You can see here in the north part, year 1 and year 2, and you can see this is for, in red here, you have the cases, the reagent cases for IgM IgM for dengue, and also chikungunya you have IgM also, Zika IgM, and IgG for chikungunya and Zika also. You know that you have many cases related to cross-reaction between antibodies. Okay, I finish. And here you also doing the microarray protein serology, but this we will explain tomorrow. I can pass here. Oh, and this you also doing the blood count for all the riversides. All the other riversides we collect the samples and give the result for also it's in Portuguese, but the glycolysis area also here. You also see the renal profile. And one thing that is very interesting is this one here. You have this dyslipidemic profile in majority of them, in the majority of the riversides. And our next step is to do more 3 years in the Pantanal region, in the same area, and also to try to start the next year in January here in the San Francisco River. This is not a ship, this is a boat. You decrease the number of people here, and also in Amazon region. The only thing that I don't have is the funding. I don't have the funding for this. I have the ship from the Navy, the Brazilian Navy, they give the ship to do this, but you don't have funding. This is in San Francisco region. That's San Francisco River region. Okay, and I'd like to thank it. And sorry for a lot of slides, but...
Thank you very much. Thank you very much, Luiz. Next talk is by Maria Ogrewalska. The title of the talk is Environmental and Animal Surveillance in Antarctic. Maria is a researcher and biologist. She holds a master's degree in biology from the University of Warsaw, as well as a PhD and postdoc fellowships in experimental epidemiology applied to zoonoses from the Faculty of Veterinary Medicine and Animal Science at the University of São Paulo. Since 2014, she has been a public health researcher at Oswaldo Cruz Institute, Oswaldo Cruz Foundation, in the Laboratory of Respiratory Viruses, Extomatosis virus, enterovirus, and viral emergencies, focusing on animal origin virus. Since 2019, she has been part of the Fiocruz-RJ project, participating in expeditions to the Antarctic continent with a focus on surveillance and study of influenza virus. Maria?
Good morning, thank you. I'm from Poland, and I think my boss from Brazil were thinking that I am going to miss cold, and just put me to this project. And each summer, in the very hot Rio, I'm going to Antarctic continent to collect samples. And today I would like to tell you a little bit about this project, because actually I was really ignorant about this. I didn't know what actually Brazil is doing there. And as there are many students, maybe you will be also interested to go there to make your research there, because this is a huge program actually, like over 40 years. So I will just show you some part what we are doing, but maybe some of us will apply in the next call, and you go there as well. Well, I am from the Laboratory of Respiratory Viruses, and actually my lab works with human samples. We are like a reference for influenza B, coronavirus, and measles and others, also monkeypox now. And my boss called me to the lab to develop the research about animal viruses, influenza in birds. But actually came pandemic and COVID, and I thought that I'm going to have my slow, quiet science of influenza, and then we have pandemic, and everything just get messy. But in 2019, Fiocruz got involved in the project in Antarctic. And why in Antarctic, actually? Why Antarctic is important for Brazil? Because actually it's, have you ever heard about Antarctic like a country? It's not a country, it belongs to nobody actually. But it's very important, like Chinese already are mapping their fields of minerals and oil, and actually this is a lot of ice, so it's also the source of water. And so there's a lot of interest, political interest in the continent. And currently, there are some countries that they signed the treaty that nobody is doing there nothing. They don't explore there. They are not commercial. They don't. You just can make their tourism and science. But this treaty is going to be over in some years, and a lot of countries, they want to have an influence. What is going on there, and to do it, you have to make the research. So Brazil is doing this as well. Brazil joined like in 1975. And have a research base, and is sending scientists to make a research. And this is what I would like to tell you a little bit. Just to show you how many research stations actually there are in Antarctic, and how many they have, like China, United States, they have a lot of stations. Even Poland has 2 stations. Brazil has only 1. But you can see how interesting it is for many countries this region. And for example, one of the oldest one is in the Vostok station, Russian station. It's from the 50s. But there are some others, the Polish, I would like to show you, but they are building a new one. And there are some bigger ones, like this one from Chile. This is like a small town already. And especially Chile and Argentina, they already, like, you open the geographic books, there's Antarctic is theirs. They even allowed to have a baby born there, because there is already, there's a Chilean born in Antarctic, so this is a citizen from there. So there's a lot of politics involved. There are some very modern stations, like this British one, or this Korean one. So you see how many millions people are spending to have something there. And Brazil as well, since they joined, they created a program, ProAntar, and it's over, like I told you, for 40 years, and to make a research. And where is located this Brazilian station? It's actually even not even in the continent. They called it like Antarctic tropics. It's on the, you can see the Antarctic Peninsula, and then you have South Shetland Islands and King Roger Island when you have Keller Peninsula. And in the 80s, there was a very simple Antarctic research station. And since 1982, Brazil started to make those operations, to go there and make research. And a lot of universities are involved in this research since then. in 2012 there was a fire and the station was completely destroyed. I think even 2 people died. And Brazil reconstructed and built a completely new station. They spent, I don't know, millions of reals. The station was completely built by Chinese. And this is very, very modern research station. I will show you later some pictures from there because it's like a luxurious hotel now. It's like a postcard from Brazil, from Antarctic. People are coming from other stations. Tourist ships are stopping by just to visit the station. It's really, really, really modern. And so just to give you a look how we are getting there. Brazil has a FAB in a National Air Force that's bringing research from Rio de Janeiro to Chile, Punta Arenas. And then from Punta Arenas, from Chile, we are crossing the Drake Passage with Navy ships. There are 2 ships designated for this Antarctic projects. The whole operation starts usually in October, and then the ships are coming back in the end of April. And this is, I think, very interesting, actually. On this picture, in the left top, on the left, this is a Brazilian ship, and the right, there's a British ship. So just to show you the difference in how much other countries are investing in research. This is the David Attenborough research ship, amazing that we have opportunity to visit. It's just that they have some. It's something really amazing. This is completely civil ship and just destined like some. They can make much more things than we, of course. So we are crossing the Drake Passage, one of the most dangerous passages. They can have some huge waves. So there's like 3-4 days of crossing to get to South Shetland Islands from Punta Arenas. And here's the station and the ships from the air view. And the station is can now have like over 60 people. It's working whole year, but actually for research, for civils, is open just in the summertime from November till March. And for the rest of the time, the Brazilian Navy is taking care of the station. There's like 17 Marines that they are staying there during the winter time. And there's a place of, just to show you some pictures, the hall, the restaurant, the laundry. There's even some place that you can make some small, simple surgeries. That is actually very important in the place when you have such a difficult access to some other hospitals. Yes, you go there, you are completely isolated. Yes, there is also a gym that you can run with the view on the bay. It's something really amazing. And there are some research labs, and Fiocruz has its own lab. You have real-time equipment. You can make a gel. You can make extraction. There are some equipment for whole molecular research. But of course, there are many other projects from other universities. They have other labs. And there's a lot of, there are some projects that they are already like 20 years there, each year coming back. And of course, the Antarctic continent is not free of problems, although there is no the people, they don't explore the minerals, or and then there is a lot of tourism, and it's rising each year, and they try to control it, but it's not really easy because it's a lot of money, and so there are a lot of ships and a lot of tourists and a lot of trash as well, and from ships, from research stations, and also there's the climate warming, and that's like there was this temperature like over 20 degrees registered in the summer in 2020. So it actually has a huge influence on the animals there. Even what is, for example, the change, there is less snow. And okay, what's the difference? There's a rain. But what does it mean that there's a rain? That, for example, the chicks of penguins, they have floppy feathers. And but they are prepared for the snow, and the snow is just covering them. It doesn't affect them. But when it's raining, the chicks are getting wet, and then it's coming cold, and the chicks can die. So something very simple can just provoke the whole populations of penguins can decline. Fiocruz entered for the first time in the program in 2019 with the coordinator Win the Grave, and then we participated in all except 39 expedition because it was cancelled because of the pandemic. And then now we just started the another one project with Luciana Trillis coordinating. And the idea is to see what is going on there. But there are a lot of labs participating together, and so we want to see what kind of pathogens there are, viruses, bacteria, fungi. And also there is another line to see what kind of biomolecules can be important for public health research. So to mention some labs involved, but not only labs, also the people from journalism, because each time we see how important it is to show the population what we are doing, what science is doing, what research are doing. Because like you see, even if you talk about vaccines, I think something went wrong, because I have in Poland, for example, so many people who are against vaccinations. So I think maybe we research close in our labs, we did something wrong, that there are so many people believing that the earth is flat and the vaccination is some chips that government wants to control us. So I think this, to show what we are doing, what our work is, it's for whole the community, not related with research, it's very important. So we involve the journalists that are writing about projects, that are writing in some other, not scientific language about our results. Just to show you some pictures from the field work. It was before pandemic, before H5 that I'm going to tell you, so we don't. We collect like the soil, we collect water samples, like you see the sea lions, and we collect what is in my interest, the feces of animals. And here you can see the three main species of penguins that you can find there. Adélie, chinstrap, and Papua. And here's like the colony of them. You see that they are not afraid. And they are actually the opposite. And another colony. As you see, this is a summertime, so there's no snow. This is actually even the green areas, the moss. And some other pictures also as well from colonies. and the other one. And there are some other species, like Antarctic sharks, skuas, petrels, and seagulls. And especially those species will be very important when I'm going to tell you more about H5. Well, and there are some other pictures of like we call in Brazil, Portuguese, fofofauna, sea lions, seals, and elephant seals. Well, and probably you heard about H5 that came from Asia, then through North America, and then for the first time to South America, and killing a lot of animals. And okay, there are some human cases that we can talk later, but it affects a lot of marine birds and also mammals. And just to remember you the mortality, high mortality of birds in Chile and Peru. Here's a very sad study from Península Valdés for Argentina, where like almost 90% of populations of no sea lions, elephant seals died. 90% of populations. What is the impact of H5 on those animals? And since 2019, we are like monitoring the influenza virus in the environment, in Antarctic environment. Besides, we are doing also some other research of other viruses, but what I would like to show you actually what we found in January 2024. It was the time that actually the first time H5 came to Antarctic continent, and we detected this in the seagull, and it was like a first time in the history H5 started to come to Antarctic continent. In this year, the another summer, 2024-25, we also went, and this time the situation was much more worse. We had some research groups in the station, on the ships, and also on the camp. To see the complication of working with animals in such a place, and we have to enhance our security protocols also to work with animals. We work with penguins. We collected swabs. We collected sample for the blood for serology as well, and we collected dead animals as well. And actually, when you see, there's already almost many dead animals there already with H5. And just to show some pictures, especially skuas are very sensitive. It was a very common bird, and now it seems that it's endangered. Almost many birds are just dying. And we don't even know if they are dying because sometimes they are just, you can see the dead birds on the ground, but many times they die in the ocean. So you should, but people who work with them, with ecology, monitoring them, they are like live 20 years. They said, they are not coming back. They are not there anymore. So it's a great impact on the populations. And the sea lions as well, seems they're very sensitive. Actually, this is the first year, so we don't know even the impact. We were able to sequence the virus, and we saw that actually we had like at least 3 introductions, independent introductions on the virus, and they came from the Patagonia, from Argentina, and also from South Georgia. And for the subantarctic islands like South Georgia, like a stepping stone for virus to come to Antarctic continent. And from South Georgia, there are the studies showing that the virus spread already on other islands where there are like abundance of sea mammals and albatross. So you see how heavy it can be to affect mammals. What we are doing with those results, we are we are testing and we are informing Minister of Health, Minister of Environment, and Minister of Agriculture. We also inform SCAR, this is a scientific committee that organized some research in Antarctic continent, and they created a project of monitoring H5 in Antarctic. You can see even the map, because there's not only us, of course, there are other countries monitoring spreading the virus. And you enter to the site, you can monitor what is going on. And we also monitor pass our data to Brazilian Navy, and we explain there how we can make better the biosecurity protocols for people who are working with the animals and with other materials as well. See, the last one. And once again, thank you very much, Luiz, for invitation and to share with this data with you. Thank you.
Thank you very much, Maria. I'm now very pleased to introduce you to Juliana Calabria. She will present her talk on surveillance of pathogens in sewage from hospitals in Belo Horizonte and Campo Grande in the wastewater and in the wastewater of Paraguay River in the Pantanal biome. Juliana is associate professor at UFMG in the field of applied microbiology to sanitary engineering. She completed a postdoctoral fellowship in the Department of Microbiology at the University of São Paulo in microbial molecular ecology and bioremediation of contaminated areas. And she did other postdoc at Federal University of Minas Gerais in the Department of Sanitary and Environmental Engineer. She works on the identification and quantification of microorganisms and pathogens, antibiotic-resistant bacteria, enteric virus, SARS-CoV-2, in water, sewage, effluent sludge, and waste samples. Her research also covers anaerobic digestion and pollutant biodegradation, anaerobic wastewater treatment, and nitrogen removal from effluents through the anomix process. Thank you very much, Juliana.
So, good morning, everyone. It's a pleasure to be here. Thank you to Luiz for this invitation. I have many slides. I'm going to show you some of the results of different projects, different researches that we are conducting here in Belo Horizonte, in the metropolitan region of Belo Horizonte, doing wastewater surveillance for different kinds of viruses and also antibiotic-resistant bacteria and also antibiotic-resistant genes. But we also have other projects in the state of Mato Grosso do Sul, where Pantanal is located. And we also have, of course, the NAVIO project, doing water and wastewater surveillance in that region. So I am a little bit afraid of Sonia about the time, keeping the time straight. So I'm going to start in English, but maybe I will switch to Portuguese to be fast. Okay, let's go. So for those that don't know what is wastewater-based epidemiology, it's a way to collect a sewage sample from the whole community and look for different biomarkers, chemical markers, and also biological markers, such as viruses, bacteria, and genes. And it's a very elegant approach because for many diseases, when we are with influenza or dengue, or even COVID with SARS-CoV-2, for instance, when we manifest the symptoms, we go to the doctor, or even we can go to the hospital. But for many diseases, only a small percentage of the patients look for hospitals and clinical attendance. Most of the patients are asymptomatic. And for this, by collecting a sewage sample from the whole community, we can detect the virus circulating in the symptomatic and asymptomatic individuals. So we can have a more realistic view of the situation of the disease in the population, because we can go, we can go down to the bottom of the iceberg. Oops, peraí. Okay, so talking about disease that can increase with climate change, we know already, of course, that vector-borne disease can increase. And how about these viruses? Can we measure this in wastewater? So this paper is an excellent review showing that actually it's feasible to detect the RNA from dengue, chikungunya, Zika in sewage, because when we are infected, we excrete a high amount of these viruses in the urine. So if it's feasible, can we measure this and detect this in sewage? Yes, we can. So there are some, actually there are very few studies showing this, because although we can detect, this detection is very, there are lots of challenges, because the RNA can degrade easily in sewage, because sewage is a very complex matrix. We have different types of substance in this, for instance, detergents and other kinds that can degrade the RNA molecule. And also because the temperature has a huge impact on the decay of the RNA concentration in sewage. So if we increase the sewage temperature, the RNA concentration decreases by 1, 2, 3, 4 days. And this has been shown by some researchers. But as you can see here, the concentration of RNA for dengue virus, for instance, is quite high if a infected person excretes in urine. So from 10 to the 4th to 10 to the 8th genome copies per day. For Zika, this is also high, 10 to the 8 copies per mil of urine per day. So it's feasible to do this detection and herbovirosis surveillance using sewage as a sample. And we have quite a few examples around the world. So there's this paper from Portugal, for instance, of arbovirus is not endemic from Portugal, but because of the tourism, and also because Brazilian people goes a lot to Portugal, sometimes they have a surge, an increase of dengue and chikungunya cases there. So this paper from 2024, they showed, they sample sewage from different parts of Portugal, more than 10 wastewater treatment plants, and more than 273 sewage samples were collected, and the detection rate, the detection frequency for dengue at that time was 25%. They measure a concentration about 10 to the 3 to 10 to the 6 genome copies per liter, and the detection frequency for chikungunya at that time there was 11%. So it's feasible, and then we, of course, we want to do this in our region. So for more than 2 years now, we have been collecting sewage samples from the two main wastewater treatment plants here in Belo Horizonte. So for those that are foreigners, this is Belo Horizonte, and we have more or less a population of 2.6 million people, and the city is located in two hydrographic basins, and so the Arrudas Basin and the Onça Basin. And why this is important? Because our sewer network was constructed using this hydrographic basin. And so here in Belo Horizonte, we have two main wastewater treatment plants. One is located here. So all the sewage produced by the population located in this area, the south part of the city, in the center of the city, Belvedere neighbor, for instance. So all this sewage is collected and then conducted to this wastewater treatment plant. And all the population located in this northeast part here at the UFMG, Venda Nova, for instance, all the sewage is collected and then treated in this other wastewater treatment plant. So Belo Horizonte is very interesting and has a very good sanitation condition. So more than 96% of the sewage produced by our population is collected and then treated in these two main wastewater treatment plants. So for more than 2.5 years, we have been collecting sewage from the influent of this wastewater treatment plant and also the influent from this wastewater treatment plant. So this means that with one sample per week, I can, and of course, doing molecular analysis further to determine the concentration of different viruses in this sample, I know which viruses are circulating in this population. This means 1.3 million people here and 1.3 million people there. And so we did this for herbovirus detection. For instance, I think I missed, okay. So we have been using different molecular approaches. One of them is the hybrid capture method for whole genome sequencing using viral surveillance panel from Illumina. And this is a very elegant and interesting approach, but also expensive. And in one flow cell, let's say, we can sequence 50 samples at one time, and then we have a huge amount of results. So here, for instance, I'm showing the different viruses that we detect with this panel. Most enteric viruses, such as adenovirus, norovirus, astrovirus, but also arbovirus, such as dengue. One sample was positive for Zika, it's here, and also chikungunya. And we were also able to detect monkeypox. So besides doing herbovirus surveillance in sewage, in the past 2 years, we have also been doing monkeypox surveillance in sewage. And, okay. So, and of course, there are different frequency of detection depending on the method that we used. For instance, for the whole genome sequence plus the sequence using the mini-ion, which is a long-read sequence, we detected dengue virus, RNA dengue virus in 24% of the samples from almost 1 year, comprising July 2022 to May 2023. And when we tried to use the RT-qPCR using the same kit that the public health lab uses for clinical samples, we were not able to detect not one sample. For Zika virus, we detect only one sample. Sorry. Only one sample, and this is current because Zika virus was not circulating in Belo Horizonte in 2022 and also in 2023. And this was this sample that was positive was from the hospital. So here we have a reference hospital for the whole state for infectious disease. And we have been also monitoring the sewage of this hospital, which is very interesting and important because we have different types of viruses there, and the sewage is more concentrated. So before the disease is circulating the whole community, we can see the signal of a specific virus in the sewage. And for chikungunya, we got a frequency of detection of almost 20% for these samples. And just to show you the number of reads that we got by doing whole genome sequence and also the long read sequencing with the sewage from the reference hospital. So these are the number of reads and the coverage for dengue, and these are the number of reads and the coverage for chikungunya. And these are the confirmed cases of dengue and chikungunya, respectively, in the community, in Belo Horizonte. So even with very low number of cases in the community, we were able to detect the RNA virus in the sewage of the hospital, meaning that we can use the hospitals, the sewage of the hospitals, as sentinel points to monitor afterwards a type of disease and the increase of the disease. Okay, this is a comparison between whole genome sequencing and mini-IO sequence for dengue and also for chikungunya. And here we have a much higher detection frequency for dengue when we use the viral surveillance panel compared to the primers and the method for the mini-IO sequence, but we saw the opposite for the chikungunya detection. And here to show you the number of cases per epidemiological week in Belo Horizonte in 2022 and 2023, just to highlight that when the number of cases were below 50 in the community, we were able to detect the signal in the hospital sewage. But when the number of cases increased in the community, above 70, above 100, we were able to detect the signal in the sewage from the community, so the sewage from the wastewater treatment plant, which is much more diluted compared to the sewage of the hospital. And for monkeypox detection, so for monkeypox, we are doing a very long-term surveillance. We start in July 2022, and we are still collecting and doing the detection. And these are results from the whole genome sequencing for 2022 and 2023. And then we got a detection of almost 20% for 10 months, even when the number of cases at that time, the cumulative cases at that time, were about 341 for Belo Horizonte. And of course, we got a much higher detection rate for the sewage from Hospital A, which is our reference hospital for infectious disease. And here are the surveillance for 1.5 years, from March to September. And for this, we were doing qPCR using the same kit that the Public Health Central Laboratory uses for clinical samples. So for this, the kit did work to detect monkeypox, and luckily we had very, very few number of cases of monkeypox in Belo Horizonte. And because of that, we also got a very low detection frequency for monkeypox. And we have been testing these clinical samples, these kits used for clinical samples at the Public Health Central Laboratory for other viruses. And we test this for 48 samples from July to March 2023. And of course, the detection frequencies are different depending on the virus, meaning that the circulation also varies with seasonality. I'm not showing this in this table, but this is what is occurring. And we also have been doing AMR, antimicrobial resistance surveillance in sewage, and these are results for 1 year of monitoring, just to highlight that the concentration of antibiotic-resistant genes is higher in hospital sewage compared to the sewage from the community. And, and we also, especially for these genes that confer resistance to beta-lactams, macrolides, and quinolones. And we also are trying, and we are doing wastewater surveillance in the state of Mato Grosso do Sul, together with the health department of the state there, and also together with the Public Health Central Laboratory in the capital, in Campo Grande. And there we are monitoring the sewage from 7 municipalities, so Corumbá, Campo Grande, Bonito, Ponta Porã, Dourados, Porto Murtinho, and Três Lagoas, located here in the map of the state of Mato Grosso do Sul, and Campo Grande is the capital, where the Public Health Central Laboratory is located. And so there, we went there and trained the technicians and the researchers of this Public Health Laboratory to process the environmental samples, to process, to filter, and to extract DNA and RNA from sewage. And afterwards, they apply the kits that they already have and the kits that they already use for clinical samples, and it works nicely for sewage. And here are the detection for different viruses, adenovirus, bocavirus, enterovirus, and rhinovirus for the different municipalities. And also here for different wastewater treatment plants in the capital over the year of 2003 and 2024. And I know that I run out of time, but I need to show some of the results that we got from the NAVIO projects by doing water and wastewater surveillance in Pantanal. So there we have been collecting samples from the Paraguay river water with the help of the Brazilian Navy. And inside the ship, we have an environmental lab where we concentrate the samples and extract DNA. And there we also, we also do qualitative determination to see if the water that the riverine population consumes, if they have total coliforms and E. coli. And we have some results. So we have been processing many samples. We have physicochemical parameters for all these samples, and we have been applying some qPCR panels also to detect enteric viruses, but also respiratory viruses in these samples. And so we collect more than 162 samples in 2024, the first year of the project, and then in the second year we have much more samples. Okay. And here are some of the results for E. coli and total coliforms determination in river water. So we do have E. coli contamination in river water, which is a kind of expected, but it still is below 100 cells per 100 ml of water sample, which means that this river can still be classified as class 1, which is a good water quality, which is regulated by the CONAMA, our environmental law in Brazil. And okay, sorry. Thank you so much for all the team, and sorry for the time. Yeah.
It's okay. Thank you very much, Juliana. Thank you very much, Juliana. I'm now very pleased to introduce my colleague, RaoniRajão. He will be presenting on impact of deforestation and greenhouse gas emissions on climate change. Raoni is Associate Professor of Environmental Management and Social Studies of Science and Technology in the Department of Production Engineering at UFMG and an affiliate member of Brazilian Academy of Sciences. He served as Director of Policies for the Control of Deforestation and Fires at the Ministry of Environment and Climate Change between 2023 and 24. He holds a PhD in Organization, Work and Technology from Lancaster University and completed a postdoctoral fellowship at the Wilson Center. He is now the coordinator of the Centro Tecnológico em Modelagem Ambiental at UFMG. Welcome, Raoni.
Thank you very much. It's indeed a pleasure to be here. And so for this presentation, I thought it would be interesting to take a little bit of a step back and understand what are the factors behind climate change and how those factors are not only linked to some global processes, such as the global warming linked to greenhouse gas emissions, but also deforestation as an important factor in terms of climate change. But I thought it was, for that, important to give a step back. First of all, because we have people from different areas, research areas here, and also people from students at different walks, including some of my undergrad students. So I thought it would be important to have a strong starting point for us. So first of all, it's important to remember that climate is actually a statistic concept. In Portuguese, it's a bit odd because we say, for instance, o clima tá bom, which means the weather is good now. But in English, there are actually two words for that. There's weather, which are the actual the situation of key atmospheric phenomena in that given moment in time. So it's cold right now. The weather is cold. But it wouldn't be wrong to say that the climate is cold. Brazil is a country with cold climate. And while in Portuguese, those terms are the same. We say clima. O clima tá frio hoje. Right? And this creates a lot of confusion because people say, well, you see, climate change is not happening. It's cold today in Belo Horizonte. And of course, that's not the case. What we have to look at is the average, how the average temperature is distributed, and also the distribution itself. What's the variance? And what we have been seeing in the recent 100 years and more clearly in the last 10 to 20 years, is that the average temperature has been increasing, even if only by 1.5 degree, but that means a lot because we're talking about the average. And also the distribution is changing, which means we're seeing more extremes, including also heat extremes. The second point which is important is that on average, the global climate has been, of course, has been changing a lot over the last billions of years. And so climate change is part of what happens throughout the world. But actually, we never saw this kind of temperature as the Homo sapiens first put their feet in the world. So the problem is not really climate change. It's how fast is it taking place, because geological linked climate change has been happening. The dinosaurs had hotter climate than we have right now. But of course, we don't live with the dinosaurs, right? We live in a different moment. Our agriculture is based on a given climate system. Our water system is based on a given climate system. And our health is based on a given climate system. So when you change that, and when you change that abruptly, of course, that means that us, Homo sapiens, are going to be affected very, very strongly. And what's causing this, right? We talk about greenhouse gas emissions and CO2 emissions. The first thing of all we have to have it clear is that this is a very tiny percentage of the air we actually breathe. It's less than 1%. But it's that tiny portion, less than 1%, made mostly of CO2, which actually works as a blanket and basically would make this planet a livable planet, even though it has a certain distance from the sun. Otherwise, the planet would be much colder. Water would not actually be as in liquid format. So that little percentage really makes a huge difference. And when you increase, since it's a very tiny percentage of what is in the air, when you increase and change the composition of that percentage of the different gases that we breathe, most of them are not greenhouse gases. Oxygen is not a greenhouse gas. Nitrogen is not a greenhouse gas. But that small percentage is a greenhouse gas. And of course, increasing that little percentage makes a lot of difference in terms of global temperatures. And so we have to ask, what's the origin of those greenhouse gases? And actually, historically, we have had certain level of emissions taking place every year, and they were mostly linked to deforestation, because that's, we as human, we basically discovered and started using agriculture in a more significant way, and that has basically meant that we cut down trees. Those trees, they actually evaporate their biomass and become CO2 through burning. But after the beginning of the 19th century, we discovered that fossilized trees and other biomass, which are basically fossil fuels, are a very good source of energy, much more efficient actually than wood, which was our previous main source of energy. And so we started digging more and more coal, more and more oil, and starting burning it. And so what happened over the years is that right now, more than 80% of our greenhouse gas emissions are actually linked to fossil fuels. And that is at a global level. But of course, every country in the world has its own responsibility. You have countries, take Switzerland, which has zero deforestation, even growing some trees, and this number actually is close to 100%. And then you have Brazil, which basically is the country in the world that in terms of absolute numbers deforests the most, and also has a very, very big herd of animals, of cattle especially. And cattle emit methane, which also a very strong greenhouse gas. So in our case, specifically in our case, deforestation is the main contributor to global climate change. But of course, it doesn't mean that if we stop deforestation and fossil fuels keep going burning, we're going to solve the problem of climate change. Of course not. But it means that in our case, our personal responsibility as citizens of Brazil is to reduce deforestation as a way to stop global climate change. So that's why sometimes you see those two discourses taking place at the same time, and it's important to separate the two. But then there is another factor that changed the climate, so changed the distribution, especially of rainfall and also temperature, which is deforestation, not as a source of greenhouse gas that contributes, that changes at the global level, but changes at regional level. And actually, it all started in the end of the 18th century, beginning of the 19th century, as the founder of geography, of modern geography, Humboldt was doing field work in Venezuela near the Amazon. And there was this lake called Lago Valencia, which made really many people curious, different scientists curious to understand why that lake, the level of that lake was going down and down and down every year. And every geologist and geographer that came over had a different theory. One of them that there was like this sinkhole that was sucking up the water to the deeps of the earth. Another one was saying that there was a connection to the ocean. But then, by observing how the rainfall behaved in an area with forest, and how the rainfall and basically also the streams of water behaved in an area heavily deforested, which was the case of Lago Valencia, which over recent decades have been undergoing a lot of deforestation. And then he reached the conclusion, which is that the native vegetation plays a very important role. I'm trying to translate here that the destruction of forests and taking away the plants and basically to cultivate the indigo. Indigo was basically, it's a plant used for gins, which was going up in this moment. So there was a big indigo plantation in Venezuela, has produced over 50 years, half a century, in the quantity of the water on the one side, on the transpiration of water on the other, basically the dryness of the soil and of the atmosphere. And those are sufficient causes to explain the significant reduction of Lago Valencia. As you cut trees that cover the top of the hills, of the mountains, men of all climates produce at once two calamities for future generation. The lack of fuel, in that case, because you don't have timber to burn, and the lack of water. And this has become, over the years, what was known as the dryness theory, and that was very influential throughout different countries. There was a lot of research basically trying to find out that link. And one of the actually key figures that was heavily influenced by that theory was José Bonifácio de Andrade Silva, which was actually one of the founders of independent Brazil, and he has actually wrote in many books and many treatises talking about the importance of forest conservation as a means to ensure rainfall and to ensure water supply. And even in our legislation, when Brazil presented actually its second forest code in 1965, in the middle of at the very beginning of the military dictatorship in Brazil, Hugo Leme, which was a professor of ESALQ, of the University of São Paulo, who actually was one of the people responsible for introducing modern agriculture in Brazil with all the inputs and the chemical inputs for that. He really did a very important job in terms of recognizing that farmers, they have to conserve forests at their own cost in order to warrant they have water for their own production of food. And it was based on this that already in 1965 it was passed a legislation to conserve at least 50% of all native vegetation in the Amazon and also in forested areas with more forest, and at least 20% in the rest of Brazil. A legislation, by the way, which keeps being respected in a very systematic way. Then in the early 70s, at that point we started to have some basically data being collected more systematically on weather stations across the Amazon. There was actually a very heavy investment in creating that kind of infrastructure throughout the 60s and 70s. And they started to see also through some new methods, which was basically trying to measure the isotopes from water from rainfall. Because the isotopes from water can tell you whether the water is coming directly from the ocean, because in the end, humidity comes mostly from the ocean, or whether the water was coming from the transpiration of the trees themselves. And actually, they found out at that moment that about half of all rain taking place in the Amazon was not coming from the ocean. It was coming from the trees of the same forest. So it was shown that there was a process of circulation of humidity starting in the ocean, bringing humidity to the middle of the continent, which made possible for Brazil to be a country with lots of rain, also in the central part of the country, where, by the way, has one of the largest agricultural production areas in the world, and making it very different, for instance, from Australia, which has a desert in the middle, or the African continent, which has a desert in the middle, or the United States, who has also a very dry area in the middle. And this has some very strong social economic impact. For instance, this is a paper we published in our group here, which was basically trying to measure how areas with more deforestation were having a stronger reduction in rainfall between 1999 and 2019. And then we projected into the future two scenarios. One scenario with a strong policy whereby deforestation was basically being withheld. And of course, some new deforested areas would show up, but in a restricted way. And then in a scenario with a lot of deforestation. And we found that actually those who deforest in the scenario with more deforestation, with weak governance, they actually get about 20 billion US dollars richer. However, the whole area, in terms of losses of productivity due to changes in rainfall pattern, would lose 180 billion US dollars. So even though the deforesters will get richer, Brazil will get poorer. So it's a classic problem of the tragedy of the commons. The climate is the commons, and we need to use it very wisely. And to make things worse, we have deforestation, and we have, of course, interfering in the rainfall patterns throughout Brazil and causing regional climate change. And we have the threat of climate change to the forests themselves. So what Salazar, also Carlos Nobre, and many others have shown that if we reach irreversible climate change, so a change in temperature above 3 degrees, the Amazon is going throughout a tipping point whereby the forest itself, even without being cut down, is going to basically be replaced by a very dry type of savanna, which of course does not play the role of circulating humidity. And this affects our health in many, many ways. So it pollutes the air because with a drier climate, we have more fires, for instance. You have more malaria, dengue, the different diseases have been shown in the previous research here, showed here, more allergic diseases, cholera, leptospirosis, the destruction and also lack of food, of course, forced migration, also health problems, especially in terms of the heat, waves, and also different disturbs of mental health, because actually people are becoming more stressed, especially given to the increases in temperature. So some conclusions. It's important to remember there are global factors that influence the climate. Of course, an increase of greenhouse gas emissions is a very important one, but climate change also happens at the regional level through deforestation. So you have those two layers, and they actually add to each other in a very strong way. And we think that it's very important to remember that fighting deforestation, reaching zero deforestation in Brazil is not only a matter of warranty, the stability of food production, but also it's a matter of public health. Thank you very much.
Thank you very much for keeping your time. And Raoni, now we can, and I will invite all the colleagues from this table to come and sit for a short round of questions. Please, Jorge, Khalil, Adriana, Luiz, Alcântara, Maria, Juliana, please come for a very short round of questions. We are actually in time, I must say. Thank you, Raoni. I would like to ask to the ones that would like to raise questions, please introduce yourself, tell your name and the institutions, and if you can, please direct the questions to the speakers that you would like to respond. Professor Jorge Khalil is gone. Okay. So, thank you very much. please, the ones that would like to talk, can you come up front to raise questions? Meanwhile, I would like to thank our fantastic speakers. It was my pleasure to be here. This session on climate change and infectious disease, really transdisciplinary panel with different views on these issues, how to solve. Thank you very much for all the contributions that you gave so far. And now, questions?
I have a question for Rajão. The other day I was watching a seminar from Atacho, and I got, well, at the end I was very pessimistic because everything we talk about controlling climate change, it seems to me that's something that's going to be very difficult to happen, even though we have all these plans and ideas to do that, and I'd like to hear your opinion. So if you have any chance, or we're going to have to really learn to deal with this situation.
Well, we won't be able to stop climate change. That's for sure. That's why, even in the language of the international agreement, we talk about mitigation and adaptation. So you mitigate something which is coming anyway. So it's not stopping climate change, but mitigating the effect, so making it actually slower and also more long-term. Because the problem is not the change itself, it's adaptation is to that change, and adaptation of our health system, our food systems, and our cities. So we need, and then that's the second bit, we need to adapt. And the kind of research being done here, shown by the different researchers in the panel, really shows that a big effort is taking place already, but we need more than that. We need, we need more money, we need more resources, we need more research, and because as the temperature increases, we have more arboviruses circulating, we have more a higher percentage in risk of pandemics taking place. So definitely, we have to increase our preparedness and surveillance.
Hi, thanks for the talks. My question goes to Maria. I would like to ask you, with the outbreak of avian flu recently, what's the perspective you think that could actually, those viruses could do, like man-to-man transmission and potential of epidemics? What's your, idea or your,
It's actually very, very difficult questions. We cannot really predict. We can just observe. This is like the first time we have a H5 in South America. Nobody even expecting this. So what we can do is just to observe what is going on with the virus. It's going good. It's adapting a lot to mammals, to our surprise. So actually we can monitor it. And what is going on, I don't know, really, it's going to stay, for sure. It's not going to disappear, as we observe. So we just have to adapt and to monitor how it's going to evolve. There are also some people talking about vaccination. It's not possible to make vaccination of wildlife. Although there are some people are trying, for example, to vaccinate endangered condors population in the United States, and I know that people are monitoring the human infections also to produce vaccine, especially for people who work directly with wildlife, like in Brazil, there's a lot of people who monitor the beaches. There's the program of Petrobras. So I think those people, those workers, vets, are really exposed to the risk. But what's going on, now we have just to watch how it's spreading, and to make genomic surveillance, I think is very essential to know how it's going and what's going with the virus.
Any other questions? If there is no more questions, I really would like... Okay.
For the NAVIO experience, so over these studies, what you like pathogen you found that we really need to be more careful, or you think is a big range of pathogen and to do this mitigation?
Okay. Bom, in instance, related to arboviruses, you cannot detect arbovirus because they just 3 days in each community. It's difficult to have people with acute symptoms in this time, but you are detecting IgM, for example, for Zika in the riverside. You are then it means that Zika is circulating in this area. You are detecting Oropush in the north part of the river. IgM also. The virus that causes encephalitis, like the east and west encephalitis viruses, that are circulating in Argentina and also Uruguay, and it means that in Brazil you have people in this area that have IgM without cross-reaction, and you are found also many cases of IgG for Chagas diseases in this area, and leprosy also in the north part of the river, and syphilis, many cases of syphilis. And you know, I think the worst diseases that they have is that they are more poor. If I pass the first year, and if I pass the second year, they are worse than the first time. The disease increases. The river the density of water in the river decreases. It's the diseases and more. I think it's this.
Each time you go there, you like treat the patients for those diseases that are treatable, like Chagas disease or syphilis, or how do you manage that?
Yeah, what I have to do is...
I would expect that certain types of disease should decrease the number, right?
It's very difficult for us to do something. What you can do is detect it, give the diagnostic, and send the results to the surveillance of the states of Mato Grosso and Mato Grosso do Sul, because they are in the project, because they wanted to see which kind of disease are circulating in this area, to be able to take decision, to go there and to make the active surveillance in this area.
Thank you very much. I think that now we are ready to close this morning section. Thank you very much. In the afternoon, we'll be back at 2 o'clock, right?
Yeah, I need to say something. First, congratulations for all the speakers. It was a really great session. And I'd like to ask you, there is going to be a presentation from Beckton and Dixon at 1:30. And they need some audience here. So those involved in flow cytometry and the students and the postdocs, I'll ask a favor for you to be here at 1:30. They really supported a big part of this meeting. So, and I thank you.
So, see you at 1:30.